Good evening. My name is Favour Otoijamun, and today I will be talking about a drug discovery research for Parkinson's disease with microtubule associated protein, MAPT, protein tau, MAPT. First of all, I would like to talk about the Parkinson's disease. It is a genetic and neurodegenerative disease that can influence the motor systems, the cognitive, and the behavioral parts. It is mostly seen in the substantia nigra part of the brain, and so far it's been associated with unusual activity of the alpha nucleic protein in the Lewy bodies, but some recent studies are saying that it could have an effect with MAPT and with as classified as tauopathy. So first of all, what is the role of MAPT? It is associated with a microtubule and it stabilizes it and might be involved in the establishment and maintenance of the neuropolarity. There are different isomers, but the most prevalent one is the 4R in the Parkinson's disease. And what happens when it goes wrong is that instead of binding to the microtubules, it will bind within itself, so it aggregate within itself, and up to one point where it tangles, and that is called the neurofibrillary tangles. And if this phenomenon accumulates and propagates to other neurons via synapses, it can lead to cell death. And so here's a better representation of it. So you see the filaments are attached to the microtubule, and then with some modifications, they detach. The microtubule also destabilizes, and the filaments will bind with each other. They will fold, they'll bind with each other up all the way to creating this tangle. So here's a video that is sadly not able to play at the moment, but it's very interesting to understanding the role of MAPT. Then there are a couple of challenges with MAPTs because with what I've said earlier, our goal would be to identify some molecules that can inhibit the aggregation, the folding within itself, and kind of a too late answer, last minute decision to stop propagating to other cells. But the MAPT protein is very unstable. In fact, there's no experimental models or crystals that show the full proteins. They're usually fragments of the filaments, and that's what researchers usually use, and that's also what I use today. But the second challenge is that the binding site is incredibly polar, and that makes it very hard to target. So today, what I did was I extracted the AOTC and the CHRE protein, so the tau filaments, and I prepared them with the BioVia discovery. And there's 13 unique ligands, and everything was looked at with PyRx and AutoDock for the bind, and also that's also where we calculated the binding affinities. Finally, I did 2D interaction maps to understand what happened at the binding sites. So these are the two proteins I used. So I used two basically because the Parkinson's fragments are very hard to find, and the ones that I did find are very new. So they're from 2023, and there's not a lot of research on it, especially not for binding affinity. So I also looked at 6HRE, which is a protein taken from an Alzheimer's patient. So I looked at 13 unique small molecules in total, and they were taken from drug banks or literary papers on tau inhibition. And they're into three classes: natural polyphenols, specialized inhibitors, so designed or repurposed clinical drugs. Now here's pictures of the docking with PyRx and with Chimera. And you can also see that the ligands bind around the cleft of the protein. So these are the results with both proteins, and they're not really that great. At best, they're maybe on the lower side of acceptable, except for the nitrolinib, which has a very strong binding affinity at -7.8. And it is actually a leukemia drug, but it is a leukemia drug, and the natural drug, which is derived from green tea, is not doing as well. Before talking about the interactions with before talking about the interactions with the binding site, it's important to know what each residues do for the protein itself. So the aromatic residues help to stabilize the stacking of it. So when two fibrils come together, it also then the hydrophobic residues help to form the hydrophobic cores, and also helps to drive aggregation. And finally, the polar residues form polar bonds networks and promote folding within itself, and also to stabilize the beta sheets, so the stackings. And here you could see that a lot of these residues are present when we talk about the nilotibin, and there's a lot of these hydrogen bonds, so polar groups, and some of the aromatic groups, which are very strong for hydrophobic groups, bonds. About the nilotibin, so again, it is used for leukemia, but there are clinical trials right now, second stage for the for Parkinson. When you look at the fluorotarcipir, you see very similar things to the nilotinib. The only thing is it's very small, but again, you see the hydrophobe structure, especially with the cysteine, that will create some disulfide bonds, which is the strongest out of all the bonds. And then you also have some hydrogen bonds here. The only thing is that it's a bit smaller than nalotibin, which is probably why it did not do as much of a good binding. For the other ones, you can see some of these residues that we mentioned, but they're either localized, like here, or they're much smaller, you'll see, than the other proteins. Now, as much as nalotinib was very good at binding, it does not go through the blood-brain barrier, or if it does, does it very poorly, and for that, it's not very easy as a drug. It is being approved by the FDA, but it is not easy to digest. It's not very bioavailable. And in contrast to fluorotarcipir, which has the second best affinity, it is very soluble, and it is bioavailable. It is very. Here you'll see that it has it's permeate, and it has high GI absorption. A little bit more about the fluorotarcipir. It is a medicine used for diagnosis of the Alzheimer's disease, and it is used in scans to locate the fibrils, but also to see if they're damaged or not. Last point, I would have loved to look at the docking site using protein ligand interaction profiler, although I was not able to locate any binding sites of any of the structures that I put in. I used 4 of these structures, and I believe that is because of the disordered structure of MAPT. So that concludes my presentation. Thank you very much for your attention. Have a nice day.